Hey, this is our DNA replication project. Hope you enjoy it. This video is brought to you by Lauren Bennett and Jenna Callow. Here are the parts of DNA replication. What is DNA replication? DNA replication is the process of producing two identical replicas from one original DNA molecule. The double helix is unwound, and each strand acts as a template for the next strand. Bases are matched to synthesize the new partner strands. The helicase SSBs, primase, polymerase, sliding clamp, RNase H, and ligase are all the working enzymes of DNA replication. There are 4 bases in DNA: adenine, cytosine, guanine, and thymine. There are specific rules that determine which bases can pair together. A always pairs with T, and C always pairs with G. Who searched for information about genetic material? Edwin Chargaff. This man discovered that DNA contained equal amounts of each base pair. Let's say if there was 25% adenine, that means there would be 25% thymine, because of the base pairing rules. That goes for guanine and cytosine also. Then there was also the Michelson-Stahl experiment. This was an experiment in 1958 which supported the hypothesis that DNA replication was in fact semi-conservative. This means that when the DNA helix is replicated, each of the 2 new double-stranded DNA helixes consisted of 1 strand from the original helix and 1 that was just synthesized. The Okazakis. The Okazakis were a family who conducted experiments for DNA replication. They led to the discovery of the Okazaki fragments, the shortened strands of DNA formed on the lagging strand. They are 150 nucleotides long in eukaryotes. There are 6 steps of DNA replication. First off, we have the helicase. The first step to replicating DNA is having the helicase start to separate the 2 DNA strands. This is so one can be copied and replaced by the new strand. Next, we have the SSBs. The SSBs, or also known as the single-stranded binding proteins, are very important. They latch onto the single strand of DNA that was just split by the helicase. They coat the whole strand. Its purpose is to prevent the strand from reannealing or coming back together. Next comes the primase. Its job is basically to create RNA primers. This is necessary to start the replication process. It builds up the RNA according to the template like the strand before. Step 4. Now in the middle of the whole process comes along the polymerase and the sliding clamp. The polymerase connects nucleotides together to form the new DNA strands. They are very accurate too. They have an error rate of 1 in 10 million. Following the polymerase is the sliding clamp. These two have to work together. The clamp holds the polymerase onto the DNA strand, so one does the work and the other keeps them moving along the strand. Step 5. The RNase H. Near the end of the process is RNase H. Its job is to remove the RNA primers that the primase put there in the beginning. It degrades them so the ligase can come and link everything together. The final step includes the ligase. Finally, the ligase is the last tool to replicate the DNA. After everything has been prepared and slightly copied already, the ligase comes along and links all the short DNA strands together to form the new DNA. The sources we used include Prezi.com, our Chapter 10 guided notes, our modern biology textbook, Wikipedia, Google, and Google Images. We hope you enjoyed. See y'all later.